Good morning, everyone, and thank you for joining us on a very important lecture. So, we have a very special guest to give us a lecture for today. And to introduce our guest, I'd like to call on Giselle Concepcion, the current president of PAASE. Giselle, kindly unmute yourself.
Greetings to all. Thank you, Kay. It is my great honor and pleasure to introduce our distinguished lecturer today, Nobel Laureate Professor George Smith of the University of Missouri, Division of Biological Sciences. Professor Smith was cited for harnessing the power of evolution through his work with bacteriophages, viruses that infect bacteria, to discover proteins with new functions. Phages infect and reproduce in bacteria, and Smith began his Nobel winning prize in 1984 when he realized that the filamentous family of phages could tolerate a great diversity of guest peptides, stretches of extra amino acids unrelated to the phage, genetically fused to one of the proteins on the outer surface of the phage particle. Using molecular biology methodology already current at that time, it would be possible to construct large libraries consisting of millions or billions of phage clones, each displaying a different guest peptide on the phage surface. I think I would just let Professor Smith talk a little bit about his work. But let me say that in the next 6 years, after the initial publication of his work in 1985, a number of labs, including Professor Smith's validated and expanded phage library idea, and it has continued to be enriched in creative ways by numerous scientists, including Professor Smith's co-laureate, Professor Sir George Gregory Winter, of Cambridge in the UK ever since. Professor Smith frequently argues that phage display exemplifies how scientific discoveries and innovations emerge from global scientific communities, not primarily from individuals. A little bit on the early life and academic ac- academic life of Professor Smith. He was born in Connecticut. And as a boy, he was fascinated by animals, particularly reptiles, such as alligators and snakes. And he would often go to the zoo and just observe these animals. And then after high school, when he went to Haverford College in Pennsylvania, hoping for a career in herpetology in mind, well, it looked like the biology department did not offer herpetology, and instead he focused on molecular biology, which he says was a much better fit to his abilities. After graduating with an AB in biology in 1963 and a year as a teacher and lab technician, he entered graduate school at Harvard University, where he earned his PhD in bacteriology and immunology in 1980. He served as a postdoc at the University of Wisconsin in Madison before joining the faculty at the University of Missouri in 1975. And it was during a 1983 and 1984 sabbatical with Professor Robert Webster at Duke University that he began the work that led to his Nobel Prize. He was named Curator's Distinguished Professor in 2000 and became a Professor Emeritus in 2015. As well as the Nobel Prize in Chemistry in 2018, he was awarded the Promega Biotechnology Research Award by the American Society for Microbiology in 2007 and is an elected fellow of the American Association for the Advancement of Science and of the U.S. National Academy of Sciences. Ladies and gentlemen, please welcome our distinguished lecturer, Nobel Laureate Professor George Smith. Thank you.
Well, kumusta, everyone? Good evening or good morning, as the case may be. And salamat to Joel Coelho for the original invitation to do this. It's a great honor for me to be able to talk with you today. Salamat also to Kathleen Avizo, who's the moderator, and to Giselle, who's just given me a very nice introduction. So let's start by considering the pandemics that have been ended by vaccines. There's the smallpox pandemic that was ended by the first modern vaccine, and that's a process that took 200 years. There's the polio pandemic that was ended by the polio vaccine in about 50 years. And now we're talking about the possibility that this new pandemic, the SARS CoV-2 coronavirus pandemic, might be ended. Some people are talking about 2 years. Is that even possible? Well, actually, barely a year after the pandemic was first detected, there are now something like half a dozen vaccines being administered to people as we talk. I don't want to mislead you. I don't want to mislead you into thinking, for example, that a vaccine will bring this pandemic to an end. Let's consider the case of the measles vaccine, a spectacularly effective vaccine. Yet it seems that measles is a permanent pandemic. I mean, there are countries today where hundreds of people, maybe even high hundreds of people per million population die of measles every year. And even in those countries like the United States, where no one dies of measles, it's necessary to vaccinate each new cohort of babies against measles in order to stem the pandemic. So we can't rely on any vaccine necessarily to put this pandemic to an end. It may, like measles, be a permanent pandemic. So there are half a dozen effective vaccine designs being used. Millions of people in rich countries have been already vaccinated. I got my first vaccine shot 2 days ago. I'll get to less rich countries like the Philippines later. But I'm only going to focus on one type of vaccine, and that's RNA vaccine. And that's even though it's not really the cheapest and not necessarily the most effective for ending this pandemic. But RNA vaccines have very attractive features. And in particular, they constitute a generic platform that can be rapidly mobilized without change to any new infectious disease threat, whether it's a pandemic or not. So I think they merit special attention. So here's our culprit. This is the SARS CoV-2 virion. The virion is the name we give to the virus particle. It's a pretty typical animal virus. It's surrounded by a lipid bilayer membrane called the envelope. That's just like the membranes that surround our cells. And in fact, the viral envelope derives from cell membrane. And protruding out of this membrane are 50 or 100 copies or so of a protein called spike, shown in red here. And the spike protein is essential for the infection process. So the infection process starts when spike binds to a receptor called ACE2 on susceptible cells to initiate the infection process. And spike plays additional roles after binding as well. Spike is the target of almost all coronavirus vaccine projects. So when spike enters the body, either because of an infection by the virus or artificially because of artificial vaccination, it induces a virus-specific adaptive immunity. The adaptive immune response is inducible. That is, it only appears when an antigen enters the body. Before the immune system is exposed to an antigen like spike, it doesn't respond to it. And it is highly specific for the inducing antigen, for spike in this case. So for example, the adaptive immune response to spike doesn't cross-react with the thousands of natural proteins in your body. And it doesn't even cross-react with the spike protein from most other coronavirus. Very specific. And the adaptive immune response has two arms. There's humoral immunity, and that's mediated by antibody protein molecules circulating in the blood and other body fluids. So among the antibodies that arise in response to the spike antigen are antibodies that bind to spike on the surface of the virus. And some of those binding antibodies prevent the virus from infecting cells. Those antibodies are called neutralizing antibodies. And neutralizing antibodies are a major way that the adaptive immune response protects against infectious diseases like COVID-19. So the other arm of the adaptive immune response is cellular immunity. And this is mediated not by proteins circulating in the body fluids, but rather by whole cells called T cells that arise also in response to spike and that are specific for spike. So among those spike-specific T cells are some that recognize other cells in the body that have spike in them. For example, because those cells are infected by the virus. And some of those cells that recognize other cells that have spike in them are weaponized to kill the cells they recognize. Those are called cytotoxic T cells. And cytotoxic T cells are another major way that the adaptive immune system protects against infectious diseases. So a conventional vaccine, in a conventional vaccine, the antigen itself, spike in this case, is introduced into the body in some artificial way. But I'm talking about here, I'm going to focus on a different design of vaccine. And this is a design in which it is not the antigen itself that's administered, but rather an artificial messenger RNA that encodes that antigen. So this is a messenger RNA that looks just like the tens of thousands of natural messenger RNAs that are in every cell in our body. So in particular, they have a special structure at one end called the cap, and at the other end, a string of 100 or so adenylic acids. Those are the A's. And inside, the RNA inside contains the spike coding sequence. And what that means is it's a sequence of 3 nucleotides at a time, they're called codons, that specifies the sequence of amino acids in the polypeptide that comprises the spike protein. So when this artificial RNA gets into the cell, it's recognized by the protein synthesis apparatus, in particular, the ribosomes. So the ribosomes and other elements of the protein synthesis apparatus assemble at the beginning of the coding sequence, and then the RNA is threaded through the ribosome, one codon at a time, and as each codon comes into place, and the amino acid specified by that codon is added to the growing polypeptide chain. And when the process is finished, then the whole thing comes apart. The polypeptide chain folds spontaneously to its natural three-dimensional form, and that's the antigen, the spike protein in this case. And the messenger RNA and the ribosome and the other elements of the protein synthesis apparatus are resynthesized. So over time, a single messenger RNA can lead to the synthesis of thousands of spike antigens. Now, if you put a naked RNA molecule into the body, it's a very inefficient vaccine. And that's because there are ubiquitous enzymes that degrade RNA in fluids outside cells. And also, a naked RNA molecule, even if it survives that degradation, enters cells very inefficiently. Instead, the RNA has to be formulated in a way that protects it against degradation and that mediates its entry into cells. Oh, I should say that the design here is that, in effect, in these vaccines, a few of your cells become factories for making the antigen. Anyway, here's the formulation: lipid nanoparticles. So the lipid nanoparticles about the same size as the virion, and it has roughly the same kind of structure. It's surrounded by a membrane. It's an artificial membrane. And inside the membrane are one or a few copies of the RNA. This is the lipid nanoparticle. Lipid nanoparticles are manufactured by a very simple process from 5 highly purified ingredients. There are 4 purified lipids dissolved in ethanol, and there's a highly purified RNA. And the RNA isn't derived from a natural source. It's produced artificially in the test tube using a purified enzyme and purified RNA precursors. And afterwards, the resulting RNA is highly purified to remove any contaminating species. So there are these 5 components. The RNA is dissolved in water. The lipids are dissolved in ethanol. And when you mix those 2 liquids in a certain way, the lipid nanoparticles self-assemble in huge numbers. And this is a sort of generic manufacturing process that's the same whatever the sequence of nucleotides in the RNA is. So it's immediately applicable no matter what the infectious disease target is. So what happens when these lipid nanoparticles bearing an RNA inside are administered to the body? Well, they enter cells. That's number 1 in this diagram. And they enter cells. The process isn't shown here, but it's a process that's similar to the way the virus enters cells. And as a result, the lipid nanoparticle is inside the cell, inside a membrane-bound vesicle. And in that vesicle, the RNA escapes. The RNA is these little red squiggles that escapes from the lipid nanoparticle and ultimately exits the vesicle, that's number 2 in the diagram, into the cytoplasm of the cell. And they're recognized by ribosomes and the protein, that's number 3, and the protein synthesis apparatus ensues, making, as I said, many copies, thousands of copies of the antigen, that's number 4, by the process I just described. So that's how the antigen enters the body as a result of this RNA entering the body. But that's not all that happens when a lipid nanoparticle enters one of these cells. The lipid nanoparticle also stimulates the innate immune system. So I've talked about the adaptive immune system, which is inducible. It's absent until it's induced by encountering an antigen. But the innate immune system is constitutive. It's always present, regardless of whatever it's been stimulated or not. And it consists of a whole bunch of sensors that sense the presence of molecular patterns that are characteristic of broad classes of pathogens. So these sensors are not highly specific at all. just very generic structures that they recognize. And those pathogen-specific structures are called pathogen-associated molecular patterns, or PAMPs. And a perfect example of a PAMP is an RNA inside a membrane-bound vesicle. In a healthy cell, there shouldn't be any RNAs inside membrane-bound vesicles, but in a cell that's been infected by an RNA virus, there's every reason to believe there would be RNAs inside membrane-bound vesicles. And so among the sensors in the innate immune system are 3 sensors inside that patrol, that surveil the inside of vesicles. And they're called TLR3, TLR7, and TLR8. And they sense the presence of RNAs inside a vesicle. So that I've surrounded those with that red box. So when those sensors sense the presence of an RNA that binds to those sensors, that triggers the formation of type 1 interferons in the cell. That's step 8 in the diagram. So type 1 interferons are proteins that are secreted from the cell, and they recruit other elements of the immune response, both of the innate immune system and of the adaptive immune system, to the site. And that kickstarts the adaptive immune response. So adaptive immune response pretty much requires that the innate immune system be triggered ahead of time. So I should say that conventional vaccines don't trigger the innate immune system. So most of them have to be supplemented with additional substances that separately trigger the innate immune system at the site of injection of the vaccine. Those extra substances are called adjuvants, but the RNA vaccines don't need those adjuvants. They're self-adjuvanting, as it's called. Not a very attractive word. I'm going to have to muddy this picture a little bit because I should tell you that in the current RNA vaccines, the RNA has to be modified and highly purified in order to block their sensing by TLR3, TLR7, and TLR8. And the reason is that if the innate immune system is overstimulated, that leads to a sort of inflammatory response which cuts short the adaptive immune response to the vaccine. So in order for there to be a strong response to these RNA vaccines, the RNA has to be modified and purified in a way to block that innate immune system. Nevertheless, perhaps there's some residual stimulation of the innate immune system because these RNA vaccines do not have to be supplemented with adjuvants. So let's briefly summarize the theoretical advantages of this type of RNA vaccine. First, there's a simple, generic, good manufacturing practice, or GMP process, from 5 highly purified components, 4 lipids plus the RNA that's synthesized artificially in vitro. There are no potentially or arguably potentially toxic components or contaminants. There's a very short manufacturing time scale, a very easy scale up. And the manufacturing infrastructure that has to be put in place for one pandemic, for example, is immediately usable for any future pandemic or other infectious disease. Secondly, there's no adaptive immune response to the vaccine structure itself. That's very important because if there were an adaptive immune response to the vaccine structure itself, that would mean that that platform couldn't be used again, probably. Because if you use that same platform for another pandemic or another infectious disease, the adaptive immune response against the vaccine structure would cut short that vaccination. So the fact that there's no adaptive immune response to the vaccine itself means that this is a platform that can be used indefinitely for any new vaccine pandemic threat. It's self-adjuvanting, as I've just described. There's another advantage that the RNA never enters the nucleus of a cell, and it's not reverse transcribed into DNA. And that means there's no possibility that there could be a rare oncogenic heritable alteration to a chromosome in one or a rare vaccinee that might lead to cancer. So in short, this is a generic, presumably safe, reusable platform that's immediately adaptable to any new pandemic or infectious disease threat. There are problems. One of the major problems of the present RNA vaccine is that they have to be kept very cold. And that's a major problem, a severe problem for administering this vaccine in under-resourced countries. And I would say that the Philippines comes under that rubric. So this is a major hurdle. Very likely, this is a problem that can be solved. There's many investigators that I've talked to that think that it will be possible to develop a way to freeze dry these vaccines so that they can be distributed at room temperature or at least at refrigerator temperature and reconstituted on site just before immunization. But that's work that has not yet been accomplished. So as I say, there's a number of vaccines on that are being used today. The two RNA vaccines, one by Pfizer and one by Moderna, are very effective. But they're very costly too, and they have this disadvantage of requiring this super cold chain. They do have the advantage of being reusable because they don't induce a adaptive immune response to the vaccine structure itself. So the others, some of them are much cheaper, but none of them have that advantage of indefinite reusability. Vaccine cost is a major problem for unrolling this vaccine in countries other than really rich countries. So I'm comparing the United States to the Philippines, which is a middle income country. So Philippines per capita GDP is about 1/20th of the United States per capita GDP. Population's about a third. The Philippines Congress has recently passed a per capita a budget for the pandemic vaccine project, and that amounts to about a little over like almost $16 per person, and that's a very small budget for in relation to the cost of the vaccine, the current cost of especially the RNA vaccines, and also considering that actually deploying this vaccine in the Philippines will be a very hard task, a more difficult task by far, I think, than in rich countries like the United States. In the United States, per capita budget is about 4 times per person than the Philippines. And even though the Philippines budget is pretty small per capita, it's still a 5 times higher percentage of GDP than our much bigger budget is in the United States. And the Philippines economically is less able than the United States to absorb this extra cost to its economy. Still, let's just accept that there's $40 per person per vaccine. Let's accept that cost. And also let's add another $40 per person to administer the vaccine under difficult circumstances. So that would amount to about $640 billion to vaccinate the world, the entire world, about 8 billion people. that is not a scary figure. Actually, if you take the 30 or 40 richest countries in terms of per capita GDP and add up all their total GDPs, this $640 billion would be only about 1.5% of their GDP, and that's something they could easily absorb. And I would go further. The United States by itself could absorb this cost. We've already spent on the pandemic 5 times that amount. Uh, and an investment in immunizing the entire world to this pandemic would be an excellent investment, even if we leave apart any morality or humanitarian considerations, even in our own self-interest. Quelling this pandemic on a global scale would be to our selfish advantage. So it's not the means that are lacking here, it's the political will. So before I go on, I want to roughly divide the genesis of a new vaccine and of any other new pharmaceutical into 2 phases. So this isn't a clean division. There's overlap between these phases, but I think nevertheless it's useful to think in these terms when we consider how new drugs come into being. So there's first a discovery and technological innovation phase. This is exploratory science driven by curiosity and professional ambition. And it's overwhelmingly carried out in academic labs with public funding. So there's discoveries and the innovations that depend on those discoveries, plus a few other things. And those innovations and discoveries emerge very unpredictably from huge global scientific communities. And that's what Giselle was referring to when she said, I keep emphasizing this. Primarily, they don't arise from individual researchers or research groups. So the other phase is development. And this is carried out predominantly by corporations driven by profit. Mostly development involves the final optimization of the innovation that comes out of discovery and technological innovation, on devising safe and efficient manufacturing processes, and in the clinical trials that are necessary to get new drugs actually deployed. So the money for this comes privately, and it comes from private sources with a promise of government-granted patent monopolies when successful drugs result. And this necessitates, of course, that these corporations acquire key patent rights, mostly purchased from public institutions or associated startup companies. And this means that the governments have to permit researchers and their institutions to pursue patent rights for the discoveries and innovations that arise from their publicly funded research. So let's just summarize the progress that's been made in the creation of an RNA vaccine for the SARS CoV-2 vaccine. So first, the discovery and technological innovation phase was pretty much finished years ago. I mean, for the last 2 decades, there have been many academic reports about the behavior of these lipid nanoparticle encased RNA vaccines. So I consider that done. But the development phase was pretty much not yet started when the pandemic hit. Now, of course, there's some development that couldn't be started until the pandemic hit. Anything that's specific for this particular pandemic, of course, that couldn't be started until we knew what the pandemic was caused by. But there were also generic issues that haven't been resolved that could have been resolved before we knew about these pandemics. And I've mentioned a few of them already. Can the vaccines be freeze-dried and distributed at room temperature or refrigerator temperature? Is there a cheaper and more stable formulation than lipid nanoparticles, maybe? Uh, do these vaccines in general induce a vigorous, durable, adaptive immunity to antigens, both antibodies to the antigen and T cells specific for the antigen? Um, these uh, are there unanticipated generic safety issues? These seem to be a priori should be very safe vaccines, but uh, time will tell and experience will tell. So these are generic issues that really could have been addressed before we knew what particular pandemic we were facing. So as a result of not addressing these development issues in advance, oh, and I should say it's not surprising that the corporations driven by profit haven't dealt with these generic issues. I mean, this would be a very risky investment indeed if you're a corporation that depends on profits, even if they would be an excellent investment for public investment. So anyway, as I've already mentioned, the RNA vaccine platforms were ready for development years ago, but patent-funded development has failed to address the generic issues that could have been answered beforehand. And that means that we've had to fund the development of these vaccines on an emergency basis. So here's an example. The US has, in a very hasty way, invested heavily in Moderna's RNA vaccine. So it invested almost $1 billion for development of the vaccine. That's almost the entire cost, or maybe all the cost of development of their vaccine. And it's also promised to buy 100 million doses at pretty high prices, more than $15 a dose. And I put down here the production cost about $4. I think it's actually more than that, but still, we're paying more for that vaccine than it takes to produce it. And we have an option, the United States does, for 400 million more doses at the same price. And yet, despite all this investment, the United States is allowing the company to keep commercial secrets, trade secrets, that is know-how, they can keep that, and they can keep patent rights to the vaccines that are developed with public money. And I should make one more comment here, is that commercial secrecy is a huge barrier to global cooperation and development of these vaccines and other drugs as well. So global cooperation is the name of the game in innovation and discovery. I mean, as I said, this is global community of scientists all over the world. And if they didn't communicate, this would be a very truncated kind of science. But that global communication is largely absent from the developmental phase of developing drugs. Can we do better than this? There are a number of economists that argue we can do much better for developing pandemic vaccines in particular, and for developing new drugs in general. And I'm going to spend the remaining minutes of this address summarizing the arguments of these economists. So governments today, especially the US government, are very averse from taxing their citizens. So that means that our government, much prefers to stimulate drug development by the promise of government-granted patent monopolies rather than by direct government spending. And that means that we pay extremely high monopoly prices for drugs in the United States. In the United States, it's estimated in 2016, we paid $450 billion for drugs, and that's $1,300 for every person in the country. And the Philippines is not off the hook. Almost all countries are obliged to pay these monopoly prices as well. So a major role, maybe the major role of the World Trade Organization and associated instruments like the Trans-Pacific Partnership, now defunct. And I know the Philippines wasn't involved in that, but I think it's really indicative, typical of this. Anyway, the major role, or at least a really major role, is to enforce honoring patent rights and other intellectual property rights in all countries of the world, no matter what their wealth is. So I'd like to also include another kind of cost of monopoly drugs. And this is the cost that represents the misery that some people experience because they can't afford those monopoly prices for the drugs they need. And when society is not willing to pay for it either. So I've put this in a kind of light blue because these are virtual dollars, sort of. It's economists attempt to attach a monetary value to this suffering of people that can't afford the drugs that they need at monopoly prices. So I don't think we should take the actual number that seriously, but this is really a real cost. I mean, and it reminds us that drugs are not just a commodity like, TVs or new running shoes. They're a public good. And effective development of drugs is not just a matter of commercial prosperity, but also of economic justice. So let's consider the alternative, which is developing drugs with public money instead of by the promise of patent monopolies. It may surprise you to learn that we wouldn't have to pay that much for drug development if we financed it publicly, and that's because companies don't spend very much of their money on actual development. Only 75 billion out of that $450 billion in 2016, it's estimated. So what would this public funding look like? Well, I think it would be modeled on the already existing public funding of discovery and innovation, like, the grants and contracts. It would be a little different. So probably it would be in the form of long-term contracts in broad areas of development in order to preserve research agility in the companies that hold these contracts. So there might be a contract for drugs against sort of broad classes of cancer, for example, or vaccines for new pandemic threats. That would be the kind of area that would be that these contracts would government contracts would cover. The contracts would require that all results of the contractor be made public immediately, whether they're good results or bad results. No patents would be allowed. And the reason for making these results immediately available is that it helps other people that are doing development with public money as well. No patents would be allowed on the resulting drugs. The resulting drugs would instantly be generic. And that means that no patents would be allowed on the results of discovery and of publicly funded discoveries and innovations either. And the performance of a contractor and the contractor's chance of getting the contract renewed would be assessed based on the usefulness of the contractor's results to society. And in that way, many perverse incentives might be avoided. And let's consider some of these perverse incentives. If you're a corporation driven by profit, don't develop cures or vaccines concentrate on lifetime treatments, guaranteeing a steady revenue stream. If you're a corporation driven by profit, it's a good idea to capture market share with Me Too drugs with no benefit to society, augmenting that steady revenue stream. If you're a corporation driven by profit, don't prepare for future pandemics and other things. That's way too risky an investment. And if you're a corporation driven by profit, hide your results. You don't want to help your competitors, especially you might want to, if you could get away with it, you might be tempted to hide your negative results from regulators if you could. These are some of the perverse incentives that would be avoided in a public system. You might ask whether public funding, this much meager public funding, would would interfere with the entrepreneurial spirit that has resulted in so many amazing drugs from the present drug industry. So think of these immunotherapies against cancers and autoimmune diseases. These are stunning advances in drug discovery. So will we be stilting this by having much less funded? Well, I don't think so. I don't think this would be harmful, because after all, there's no shortage of entrepreneurial spirit among the scientists and engineers that do discovery and innovation. And I don't think there's any shortage of entrepreneurial spirit among the technical people in the industry that are actually responsible for drug development either. I mean, I'm speaking to scientists and engineers, and maybe I could get your opinion on this. Do you really think that if discovery were developed by people like you and me in a corporation using public money, we do less of a job, we would be less motivated? I personally don't think so. Well, anyway. So of course we'd have to still pay for the drugs, but we pay generic prices. And that generic price is much less than the monopoly price. It's estimated that in 2016 would be about a fifth of what we actually paid for drugs at monopoly prices in 2016. So if you subtract, and the reason is that the drugs are all generic. So any company is free at any time to compete in the market to produce and sell the drug. So if we subtract the red expenses from a public system from the blue expenses from a monopoly system, we get the net saving to society, and that's in this fifth green bar. I've colored the bottom part of the bar with a light green, and that corresponds, that's the same number as the light blue bar. So this is meant to represent the misery that would be avoided by people being able to buy drugs because they're not sold at monopoly prices. And of course, these are virtual dollars, but the upper part of the bar are real, actual dollars that would be saved by funding development publicly. So this would be the money that goes to patent lawyers, to lobbyists, especially to profits to wealthy investors. And of course, the patent lawyers that would lose their job, and the lobbyists would lose their job. That there are some people, those are real economic costs, but I have a hard time thinking of the loss of those profits to investors as an economic loss. Maybe we should even think of them as an economic gain instead. I mean, actually, patent monopolies, not just in the drug industry, but in general, is one of the important ways that wealth is funneled upward to the richer and richer people, and responsible for, um, uh, partly responsible for the obscene wealth inequality that characterizes so many economies today. So that's it. That's the vision for a different way to develop drugs. I don't actually think that these economists have developed. I really don't think this is very visionary. I mean, it's based very solidly on the way we already fund discovery and innovation with public money. It's it's hardly anti-capitalist. After all, the development would, this envisions the development still being primarily done by profit corporations, but they would be in the terms of contracts rather than um, in the view of patent monopoly pricing. And also, I think this is a a mode of development that is more in keeping with our common notions of economic justice. So thank you very much, and I'd be glad to take criticisms, questions, uh, whatever.
Thank you very much for that, George. Yeah, so I'd like to invite Joel Coelho, yeah, to
All right.
give the first question.
Okay. Thank you, Kathleen. And thank you, George, for your excellent lecture. I thought it was meticulously elucidated and cogently argued. But I want to explore a little bit more about the second half of your lecture regarding monopoly funding for pharmaceutical companies. Just to put it in a proper context, for instance, there's about 300 plus industries in the United States, and about 75 of those are considered IP intensive. And of course, the pharmaceutical industry is part of that. And those 75 IP intensive industries or sectors are responsible for over a third of the US GDP, and they create about 45 million US jobs. And then also, if you look at different countries in terms of their pharmaceutical exports, the United States is ranked number 4. Number 1 is Germany, followed by, I believe, Switzerland, Ireland, and then the United States. So one point is, and I really agree with your contention here, but it would seem unrealistic for the United States to act on this unilaterally. And also, in terms of, are you suggesting only removing the patent monopoly for vaccine, for pandemic vaccines, or for all drugs? So if you could elaborate a little bit further on that.
Okay, that's a really good and challenging question. um, well, of course, what I painted was a picture where ultimately a lot, maybe the great majority of drug development would be carried out with public funding. And of course, some of it already is. I don't actually think there would be that much resistance to vaccine development, especially vaccine development in preparation for future pandemics being carried out with public funding. I doubt, in light of what was experienced over the last year, that there would be much resistance to that. But I actually don't think there's going to be a huge difficulty to, harm to the economy from this. I mean, the technical staff will still have to be employed, maybe more employed than they are now when with public funding. I mean, there's some staff, as I said, like the patent lawyers and the lobbyists and so on, that they would have to find new work. But I think the job situation would be very much the same. And also, you're talking about exports. Well, okay, we would be exporting drugs. We export knowledge. Maybe the knowledge would go out for free. It might be in our selfish benefit to just make the knowledge free, even if no one else agrees to this. but we could make arrangements, and I think most of the economists that write about this think in terms of global agreements among countries to sort of share their knowledge in this way. But we would still be exporting drugs. We'd just be exporting, generic drugs, and probably more likely we'd be exporting the wherewithal to make generic drugs in other countries. But I doubt that this would actually hit our exports. I think our exports would remain pretty similar to what they are. They would just be in a slightly different form. But I think even if, sure, we probably will go fairly gradually. We will go, as a practical matter, fairly gradually to this kind of funding. And we already are sneaking up on it. More and more of the drug development is being done publicly. But we'll probably sneak up on it, but I really don't think it will be that disruptive economically. But I'm not an economist. I just read economists, and I play one not on TV, but on Zoom.
All right. No, but that was really insightful. So thank you. Much appreciated.
Thank you. Yeah, now before I go on to the questions on the chat, I'd like to call Giselle. Giselle?
Thank you, George. I think you ended your talk with very important issues. Uh, Personally important to me because I'm in the field of marine drug discovery and development, and I collaborate with US scientists. And we are always challenged of how to bring our discoveries of compounds against cancer and infections and neurodegenerative conditions to the next level of development. But I won't talk about that now. Instead, I'd like to ask you, George, are there models in the world that you could follow regarding use of generic drugs or development of those, such as India? So 15 years ago, I know that India was not recognizing intellectual property or patents, but more recently they decided to do it as well. But in the Philippines, a lot of the drugs come to us now in the generic mode, and we know that is after how many years? 2 decades of patent protection. Okay, there's another model, but then I don't know if it has survived. And this is from the European community, the European Commission. There was a group of European countries that was calling for drug candidates that would met certain criteria, which would then or maybe could be developed by the European Commission or European Community. So can you see such kinds of arrangements between a government and scientific communities worldwide to move forward the development of vaccines and drugs that would be affordable to countries like the Philippines? Thank you, George.
Yes, so you point out that there are some cases where patent rights and other intellectual property rights are not recognized. A really famous example is in the HIV drugs. So originally, the companies that developed those drugs were not willing to sell them at monopoly prices in South Africa and other places which were the center point of the AIDS global epidemic. But eventually, those companies came to realize that it's to their benefit, actually, to make those drugs available at generic prices in Africa and elsewhere in the third world, because that allowed them to continue to charge really high prices for the drugs in the first world. I'm not sure that's a really great model to follow. I should really ask you, you worked in this industry, but let's say that you worked and there must have been tremendous pressure on you for that drug to be successful. So let's say you worked in that industry now, but what's required of you is to do really good, solid science. And if that solid science results in a conclusion that this lead that you've been following is not a good lead and pretty surely should be abandoned, you would still be a successful scientist. Would you like to work in an industry like that? In an industry where the your whole livelihood and your reputation and so on within the company doesn't depend on the ultimate success, which is kind of rare in the drug industry. I mean, of all the drugs that are developed, just a few percent, 5%, I don't know what, actually end up in something that can be sold. But let's say you worked in that same industry, but your progress, even if it doesn't result in a saleable drug, is still recognized and you still have your livelihood. Wouldn't that be a nice place to work?
That would be ideal, George. Well, U.S. government tried to link up academic institutions with drug companies. Okay, so there were some programs funded by the National Cancer Institute and the U.S. NIH, and there have been a few positive outcomes of those linkages between academic groups and pharma.
So there's also Canada. Oh, sorry. There's also Cuba is another model. I don't know if they recognize any intellectual property rights at all, but they have a pretty active drug development industry. They don't sell internationally very much, at least not that I'm aware of. But healthcare in Cuba, not a rich country, is pretty successful. So that's another model we might look at. That's not the model that I put forth, and it's not the model that that at least U.S. economists that write about this subject have in mind. They're still, capitalist model. It's just that the work is done by means of contracts. It's funded by contracts rather than by the guarantee of patent monopoly rights.
Thank you, George.
Okay, the next question comes from Fabian Dairit. So it's in the chat box. What would you advise the WHO?
The WHO?
Yes.
To let my country back in. So the WHO, I don't have really technical advice, and I'm not that deeply knowledgeable about the WHO, but it's an example of global cooperation that I think is really, it would be a huge advantage for drug discovery in general. Not drug discovery really so much as drug development in general, because drug discovery is already international, but drug development in general. And of course, that's particularly the case in emergencies like pandemics. If the WHO had been, we have this, what is this, COVAX, this sort of global cooperation for vaccine development. It's not very strong, and it doesn't include most of these really high-profile drugs like the two RNA vaccines, like the two RNA vaccines. But if that were more universal and it was efficiently administered by a global power such as the WHO, I think that would be an advantage. The WHO, there's lots of criticisms of it, but it is a existing global organization that I think would be a natural way to be kind of the headquarters for global cooperation.
Thank you for that. So I'll take one question from the YouTube. So there's a chat. This one comes
YouTube,
from
one of my favorites.
Yes.
my friend. YouTube, right?
Yeah, so one of our viewers from YouTube, Ramil Arshosa, is asking, do you think there would be an impact with regards to the efficiency of the vaccines, depending on the type of brand? And maybe if we roll out, for example, a more, if we try to remove patents, for example, or develop more public funded vaccines, do you think that would have an impact with regards to efficiencies?
Well, it's not clear to me whether he's asking for efficiency of development or with the efficacy
Or, yeah,
of the
efficacy. I think more on the efficacy.
efficacy.
Yeah, yeah.
I don't see why that would be different, actually. I mean, presumably, I mean, whatever drug it is or vaccine, whatever it is, it still has to be licensed. That means it has to go through trials, and the trials would be set in a pretty strict way. So I don't see why efficacy would tend to be less, whether no matter how the drugs were developed. I might be missing the point of the question. I'm sorry. Do you? Uh,
Well, actually, it's more like does the branding affect the efficacy of
Does
the
the
vaccine?
branding?
Branding, yeah, branding.
Well, I don't see why that would be true. I mean, if if you have a biosimilar or a the identical generic drug going through the same pretty strict regulation process, I don't know why it would be generally lead to less effective less effective vaccines or other drugs. I don't know. Maybe I'm still missing the point, Kathleen. I'm
Okay.
sorry.
Anyway, it's fine. Okay, so the next question comes from Candy, who's joined us here at Zoom. Can we use this technique for a predictive production of vaccines? Is there merit in thinking of a predictive development of a vaccine?
So you said predictive.
Yes,
Is
predictive.
that way?
Yes.
Well, okay. I was trained in immunology. Of course, this was in the early 60s, so I'm a little out of date. But I, um, it seems to me that that vaccines have to be they have to be very close in structure and sequence and so on to the actual pandemic threat.
Hmm.
So it's hard to think that we would be able to predict in advance, for example, a upcoming new coronavirus pandemic. That seems unlikely to me. I mean, we know broad classes of coronaviruses,
Hmm.
which are a good candidate for a new pandemic. We know broad classes of them, and so we recognize the classes almost instantly, but that's not enough to develop a vaccine. I mean, the SARS and the new SARS CoV-2, they are the same family, but
Hmm.
the vaccine against one is not going to be effective against the other. So I don't
Yeah.
see much room for sort of predictive
Hmm.
vaccines.
Okay.
But I would say that RNA vaccines are predictive in the sense that the platform for the vaccine, not the actual structure of the antigen, but the platform is reusable. And in that sense, that is a predictive, you could say predictive, preemptive
Hmm. Hmm.
a form of vaccine development.
I see. Okay, thank you for that. The next one comes from Mario. Can you please elaborate on the relevance represented by Cuba as far as vaccine development and distribution? Cuba as an alternative model.
Oh, I wish, uh, Mario should answer that question himself. I'm really sorry, Mario. I just don't know enough to answer that question. And I strongly suspect that you do know enough to answer that question. So if you're still on,
Mario, are
could
you
you
there?
address
Yeah,
that yourself?
Okay, yeah, he's here.
uh, I'm here. I'm actually driving back home. So uh, I mean, I think so. I want to know from you kind of a more of a technical side of it, because I know a lot about Cuba in terms of kind of the broader political spectrum, but I haven't really never looked into the science deeply. And you know, so anyway, so it's my impression that Cuba actually has an incredible viable model in terms of vaccine production and distribution, not just at the moral and ethical level, but also implemented in a way that actually is successful. I mean, you already mentioned that the healthcare system is remarkable, but I just I mean, I don't know I don't have the scientific knowledge, so I'm not an expert in that regard. But
Oh,
I do think so. I do think so.
looks like
That's my
I need to do research. I wish I had the suggestion earlier so that I could look up on that. So it looks like that would be a fruitful thing to look at. Whether that really is a really good vaccine development industry there. Certainly, their rollout of vaccines and other medicines is it's really a remarkable model for the rest of the world, despite the fact that it's operating under tremendous economic stress, for example, from the US economic boycott.
Yeah, and if I want to, my add, so if I wouldn't say close friend of mine, but a friend of mine was the only member on the team, an only foreign member who happens to be Italian on the Cuban team that developed the vaccine. So I had a conversation with him, and he told me, great things about the model and the way they operated. So anyway, I would love to hear more from you, George, because I'm sure
Yeah, so
you understand much better.
You'll email me his contact so I can contact him.
Okay, great. Absolutely. Okay.
Okay, and I hope you can give us updates as well on that. All right, I'll take a couple of questions from the YouTube chat. So thank you so much for your excellent talk. This comes from ML. Can you give us an idea what would be the minimum loss to be passed to nudge the current system that you were proposing? Also, wouldn't the enterprise value be badly disrupted? He also had a follow-up question. If you can share your views of what the transition period from the current patent-driven to a more public interest-driven system would look like.
Boy, okay. Is this in the chat? Is that right?
Yes, it was in the chat. In the YouTube though.
Oh, in the YouTube chat.
Okay,
Okay.
but I can put it here if that would be helpful.
The problem is I didn't get everything.
Okay, maybe we can begin with his last question on how do you view the transition period to be like from the current patent-driven to a more public interest-driven system for development.
Yeah, so that's related to what Joel asked.
Mhm.
And as a practical matter, it's going to be really a continuation of a trend that's already existing. That is, we are already increasingly developing drugs, vaccines, and so on with public funding in a manner that's similar to what I described. And different from, for example, the emergency development that I described for Moderna, which allowed the company to keep secrets. That I think is inimical to this. But I think we are already in the process of increasingly funding drug development with public money. It's just that it's a slow process because it means more taxation, more public spending. But I think we spend something like $40 billion today in the United States on discovery and innovation in biomedical. And so the $75 billion wouldn't be that much more, and we will just have to go up gradually, I think. I think it will be gradual. And we get the low-hanging fruit, the things that are more obviously prone to public funding, and that would include vaccines, I think.
Okay, thank you for that. So, yeah, Al, maybe I can ask you to unmute yourself to ask your question about the models of China and Russia. Al, are you there?
Yeah.
Yeah, okay, thank you.
Nice proposal, Dr. Smith, on your here in the Philippines. We've been working for private partnerships where we involve both government, academe, and private sector. Not just specifically the foundations, but even the actual companies themselves pay for the vaccinations. Hopefully, we can extend that towards development as well. From since we don't have a very strong patent base, most of the patent coming from our actual reform base, definitely admit the model of India where they rejected it first so they can do their own generic. And once they made money on generic, then that's when they start to do a capitalistic model like the US. But my question would be, there is clearly the Sputnik and the Chinese Sinovac vaccines that are, if you can think about it, as mainly public. And this is where it also ties in with their regulatory process and pathways, which is kind of a black box for us. How do you envision this model in the US that you're suggesting of having more public development affect the regulatory as well as the eventual partnership with the private sector again? Because you're removing them right now with the public development, and then you're going to bring them back in here in the Philippines. At least you're going to bring them back in to help in the eventual dissemination or paying for the actual purchase of the products.
Mhm.
How do
Well,
you think that whole system works? I know it's a bit a convoluted question, but this is the situation that we have in the Philippines already. And we're trying to battle between the US model, which I'm a product of. I had a company in the US for 20 years and I benefited from the patent and prosper. And now, yeah, I'm in with you in the sense in the model of the public development as well. That's happening now in the country. And I'm also proposing that, except that the private sector is still in need of a model that works. So, and again, the regulatory environment here is also quite, I would say, not so friendly and not very sophisticated and developed. That is another blocking to the actual impact of such a model.
so let me ask, you're talking about the regulatory atmosphere in the United States? Yes, okay. All right, so in the United States, and this is, I think, pretty much the same in the European Union as well. The regulatory apparatus it's supposed to be independent of the actual drug development, even if the drug development is done with public money. And even if it's done, for example, at the National Institutes of Health, which is a government institution. And I think that's pretty necessary to separate out the process of approving drugs, vaccines and drugs in general, from the actual development, because there's conflict of interest, even if it's not primarily economic conflict of interest. And I honestly don't know how the approval of drug works in China. I'm sure it's government, just as the development of the vaccines are public as well. But I do know that virtually everyone that writes about using Chinese vaccines emphasizes that they need to go through the US or European regulatory process. We're not accepting the Chinese process, and probably they won't accept ours either. I don't know. I mean, most countries are very jealous of their regulatory process to make sure that it's right, that they agree that this is something that should be sold. So I must say I'm not that familiar with how it works in China. I mean, they of course were right at the start of this pandemic, and they're the ones that released the first sequences of the virus, which allowed the almost instantaneously production of an RNA vaccine. Because I mean, it was like 19 days between getting the sequence from China and the first vials of vaccine coming out, not for putting into people, of course, but for, you know, like putting into mice, probably.
Okay, thanks.
So,
Um,
thank you.
all right. So the next question comes from Teresita de Guzman. Is there a danger of developing severe hypersensitivity reactions or immune tolerance when an mRNA vaccine becomes a manufacturer of the spike protein?
So, so so the question is an inflammatory response, and what was the other one?
Immune
Allergic
tolerance, either
immune
hypersensitivity or immune tolerance.
hypersensitivity. Okay, you can make it's rare, but you can have a um, adaptive immune response to RNA itself. These are pretty small amounts of RNA that are introduced into the body, and they don't last that long. But that's a possibility. I think there there have been papers pointing out or arguing that there is a slightly higher level of like anaphylactic type responses to these vaccines than from most vaccines. Almost all vaccines in a few people generate a anaphylactic response, and if it's a real anaphylactic response, it's an emergency, and that's why when you get a vaccination, no matter what the vaccine is, you sit around for 15 minutes after the vaccination, and the staff are there with EpiPens or some other way of administering epinephrine to quell an anaphylactic response, which can be deadly. And it's not clear, but it possibly is true that this is a little more common in these RNA vaccines than in other vaccine designs. I think we really don't know that, but there have been articles that are arguing that might be the case. I should point out, though, that millions of people have already gotten this have gotten this vaccine, and I don't know of any reported cases of a really serious life-threatening type of anaphylactic reaction, but there may have been ones. I don't know. They probably would get into the New York Times instantly, so I kind of think that I wouldn't know about them.
Yeah.
Maybe someone else has heard about this. But the tolerance, okay. Yes, all right. So it is true that under certain circumstances, administering an antigen induces a kind of adaptive immune response that's against actually making antibodies or having immune T cells, that actually works against that. And a perfect example of that is your own proteins, the antigens that are in your own body. You're tolerant of those, and that's because of a negative kind of immune response that turns off and quells in a couple of ways the elements of the adaptive immune system that respond to your self proteins. You're tolerant of self, so that is possible under some circumstances, that an animal and a person can be made tolerant of things that are not actual self antigens. And I guess that's a remote possibility. I haven't heard of that as being a concern with these vaccines at all. And I don't know why it would be particularly a concern for the RNA vaccines, but I don't know.
Thank you for that. Actually, a lot of the questions are asking about the hesitance with the mRNA vaccine. So one question comes from Fabian Dairit asking, how do you think can we reduce this hesitancy towards being vaccinated?
The antipathy towards the vaccines?
Yes.
Yeah. Well, I don't know. There are some countries like France where polls have shown that like 50% of the people are not willing to get vaccinated. I think, in all vaccines, that resistance goes down as more and more people get the vaccine, and people tend to know people that have gotten the vaccine and haven't had a bad reaction or anything like that. Um, um, and I don't know, do people, how about other people's opinion? Does it seem that there's particular resistance to this vaccine,
Um,
to any of these vaccines against the pandemic, or is this just like the resistance or antipathy to any vaccine?
Okay, I've heard some of my friends like talk about hesitation with this new kind of vaccine, particularly because of the new way the new way that it's an mRNA, quite different from the more traditional ones. So I think
Mhm.
I've heard some of my friends, who are actually doctors as well, who have mentioned that they wouldn't want to take it yet, and that they would wait a while before getting administered with this vaccine. So yeah.
Well, um, if if uh, if it really takes like 5 years or whatever to immunize everyone, to vaccinate everyone in the Philippines, well, they'll have plenty of time to look at other
Do it.
people who have gotten the vaccine. I'm not sure that's a good thing, but yeah. I mean, it's kind of natural. I mean,
Mhm.
I'm probably reckless in the other direction. I would say I would immediately accept
Yeah. Yeah.
taking an RNA vaccine, just because it just seems like such a promising technology because of its reusability, its
Mhm.
indefinite reusability. But I'm kind of like maybe I'm making an irrational decision in the wrong direction. And of course, there is that question that overstimulation of the innate immune response that I mentioned. I mean, that's certainly something that researchers didn't anticipate until it was actually demonstrated. um, it was not anticipated that RNA getting into it was anticipated that RNA would stimulate the innate immune system, but it wasn't anticipated that stimulation would be harmful to the vaccine, and it turned out to be. So, there's always things that you don't anticipate, that we, we can't know the future perfectly, and we just can guess about it. We can make educated guesses about it.
Yeah.
Anyway, I would take it in a second, and I did.
Yeah. Yeah. So I think Giselle would like to comment.
I think, George, this talk is very important
Yeah.
to scientists and the general public in the Philippines and other developing countries to try to explain why mRNA vaccines are safe.
Mm-hmm.
And it's basically the science of it. Unfortunately, here, as well as in the United States, there are what you call anti vaxxers.
Yeah.
And you'd be surprised in some medical hospitals in the United States, including the one where my daughter is, 60% of the medical staff are vaccinated. The other 40%, the non-MDs, are not, have chosen not to be vaccinated because they don't believe in vaccines. But anyway, also in relation to anaphylaxis and all, I've posted in the chat a review article in the New England Journal of Medicine that is co-authored by a medical doctor from Vanderbilt University to further explain what George is trying to say, that there seems to be minimal anaphylaxis, or there are always outliers, as we might say.
Mm-hmm.
So thank you so much, George,
Yeah.
for this
Yes, I
important talk.
I I read that article, and that's the one actually I was kind of referring to. And it got into the New England Journal of Medicine, which is a really respectable journal. So I would say that this is this has to be looked at. Absolutely, we can't know this in advance, and someone like me that is like super willing to take this vaccine may be acting foolishly, but I don't actually think so. And a priori, this is a vaccine that just has these small, highly purified molecules, the lipids plus the RNA. It's hard to imagine these as being harmful, except possibly the RNA.
Yeah. Okay, I have another question from the YouTube channel. So this one comes from Pros Naval. So thank you so much for your enlightening lecture. Could you please elaborate further on the importance of publicizing or even publishing negative results? What do you think is the proper avenue for this?
So the question is, what's the importance of publishing negative results?
Yes.
And probably some of you are scientists who have tried and failed multiple times to publish negative results. It's really hard to publish them. And so I think that this is different from posting or making available negative results in the context of drug or vaccine development. So that means that that because that kind of negative result that's very specific can be extremely useful and would be instantly taken up and looked at by other people. So, for example, if you are working on that business of freeze drying the vaccine so that they can be distributed at room temperature. Okay, so let's say you're a company doing that, and there probably multiple companies are independently doing that and not telling each other what they're doing. So they try 5 different methods, with different compounds to stabilize during the freezing process and so on. All those things, the standard methods. And they show their work and it's clear that it doesn't work. That is enormously useful to other people who are trying to accomplish the same thing. Okay, I'm not going to try that method. Let's think in a different way. That's those kind of negative results during development. You know what they're like? They're like negative results in the lab, in a regular research lab. You get negative results all the time, and your colleagues, the other people in the lab, find out about those results instantly, and they say, yeah, we got to let's go back into the conference room and figure out another way to do this. I mean, they're really useful in a practical way. It's just hard to get them into formal peer-reviewed journals to get negative results in that. And so maybe that's a good reason to bypass peer review. I'm not sure peer review is necessary anymore. I'm for just like posting everything. The internet, you can find anything that's been posted, and other people can say that's BS, you're wrong, or I don't believe it, or whatever. Maybe that's a better way to distribute our knowledge and our findings and our ideas rather than going to peer review journals. Anyway, that's another of my hobby horses.
Okay, thank you for that, George. And I think this might be our last question. Can you comment on the issue of equitable vaccine availability or distribution?
Equitable distribution. That is really a key question. So of course it's a key question of like what countries get to have the vaccine, and I'm from a country that's kind of like the number one selfish country in this regard, and we've bought up enough vaccine theoretically to immunize or vaccinate our population several times over. And it will be a long time before we're going to share that vaccine with any other country. We certainly don't actively work on that. And I mean, I think it's just a moral thing that, I mean, I hate to be in a universe where everyone acts like that. I just think that we'd be in a more satisfactory world if we were all, concerned about other people. But like more to the point, in this particular case in vaccine development, for a pandemic, I mean, really, none of us is safe until everyone is safe, as as people say. I mean, really, if we are to quell this pandemic, even if it's a permanent pandemic like measles, we have to do it the world over, because a virus doesn't respect any borders and so at all, and easily, unless we close all our borders, which would be enormously damaging to us and to everyone, unless we close our borders, we're going to get any pandemic that's around there is going to enter our country, willy or nilly. So I think that it's just in the case of vaccines, especially, like, selfishness is really a bad policy, even from a totally selfish point of view.
Thank you for that. So, Joel?
Yes. George, this is just a quick science question. So how do we know really that the mRNA that's delivered into the cytoplasm cannot enter through the nuclear membrane and then reverse transcribe into DNA that could be spliced into the chromosomes? How are we sure that that's not going to happen?
Well, RNAs have been studied for a long time, so I don't think anyone has has got evidence that happens. That RNAs end up in the RNAs that aren't reverse transcribed and end up in the nucleus. And also, we don't have natural. I mean, we do have some reverse transcriptases. I mean, and of course, they're viral reverse transcriptases. Actually, our genome is filled with remnants of retroviruses, many hundreds of them, maybe thousands of them. And some of them I'm not sure that this has been demonstrated, but some of them may have still the reverse transcriptase gene in them intact. I don't know. I'm not sure that it's certainly rare, but it's possible. But then to ask this RNA, this reverse transcriptase, to have the conditions that would allow it to reverse transcribe this RNA is a pretty tall order. So we never say never in science, as I'm sure you know. We never say never, but it's pretty wildly unlikely. And this is different from, for example, the adenovirus-based vaccines, where the adenovirus DNA, it no longer acts like a viral DNA, but it bears the DNA for spike that gets into the cell, and so there's much more of a chance there. That still a really low chance, but there's more of a chance there that by some random mechanism that will integrate into a chromosome and inactivate something or activate something that leads to an oncogenic mutation, a cancer-causing mutation or cancer-promoting mutation. That's conceivable, more conceivable with a DNA vaccine than with an RNA vaccine.
All right. Thank you, George.
Okay, thank you very much. I'm really sorry. I think we have more questions, but it's already 9:30 and past 9:30 in the morning, and we need to close this particular session. Thank you very much again, George. Do you have any final words for our audience?
No, thank you. And I don't know how you say goodbye in Tagalog, so maybe you can tell me.
Paalam.
Just
So
say
I've learned
hasta la
three
vista.
words.
Hasta la vista.
Maraming
Right, see you
salamat
next
po,
time.
George.
So one of the words comes from Spanish, one comes from Arabic, and I don't know where goodbye comes from.
Gracias, paalam. Gracias.
All right. Thank you so
Yes.
much, George. Thank you.
Okay, thank
Okay,
you
goodbye.
very
And thank
much.
you
Thank
very
Thank
you
you
much
very
so
for
much.
much,
hosting.
everyone, for joining us this morning. And if you want to know more about the activities of PAASE, please log on to paase.org, which is our website, official website. And we'll be posting the recording of this particular video in our YouTube channels, PAASE webinars. So thank you again, everyone. Good evening to those of you in the US, and have a great day for those of you in the Philippines.
Thank you very much. Yeah, thank
Bye,
you
Thank you
all.
guys.
all.
Thank
Awesome. Great.
George, thank you so much. Just